Welcome to another episode of Data Skeptic Animal Intelligence. I'm joined as always with co-host Becky Hansis-O'Neill. Welcome back, Becky.
It's good to be here, Kyle.
So today's a really interesting one. We talk about a lot of data collection and some novel ways to measure animals in the field. What are listeners in store for?
I'm so excited about this, and I'm glad we found an ecologist that's doing this. So many animals really hard to observe. Either they're nocturnal, they live in a burrow, they live in the ocean. We just can't tell what they're doing. So some folks have had really smart ideas of putting different data loggers on these animals to create machine learning training sets. And if we look at those animals in the lab and video them, we can match up the data from their data loggers with the behaviors they're actually doing. And if that's accurate enough, we can create a really good training set, throw these data loggers on wild animals, and then come back and make some pretty good guesses about what they're doing. And Ryan's doing that with kangaroo rats and rattlesnakes.
Yeah, he's literally out there with colleagues, I presume, in the wild, sort of gluing these little apparati on these creatures and then following them around. It's kind of interesting.
Yep, the kangaroo rats get cute little backpacks.
Yeah, and much like I just got my first Apple Watch, so I'm learning all about the sensors and the tracking it's got. I assume this is a little bit more sophisticated. I know they have a better price point on their apparatus, but tracking a lot of deep data too that gives us, I think, a unique insight into animal behavior that historically just hasn't been there.
Exactly. I think this is one way that machine learning is really propelling behavioral ecology and conservation ecology forward.
Yeah, the field found a way to collect a lot of data and needed a way to process it. So hopefully ML and the data collection can come together in a nice way. Do you have any aspirations to put similar trackers on the species you study?
When I read about this, I got really excited because I study tarantulas and some of my study has to do with wild tarantulas. And we're talking about animals that don't need to move every day. So, knowing what they're doing, how often there's a feeding event, if they're coming in and out of the burrow, if, say, living next to a wind farm is, say, changing their behavior versus living near a trail versus being out in the wilderness, all these conservation questions could be answered with some of the same things that Ryan is using, provided I could get data loggers that were small enough. So I would love to start using this. This is amazing to me.
Is there an alternative, like somewhere between being like the Jane Goodall of tarantulas and spending a few weeks observing versus putting a tracker on? Is there any source of data today?
There is. And I know a guy, another graduate student working on this, and he puts cameras out in front of burrows. He's looking at maternal behavior of mother tarantulas. And yeah, it's the same problem that folks have heard multiple times through lots of our interviews is you have to go through all this video data and try and pick apart these valuable little pieces of information. So it exists, but it's really tedious.
And how practical do you think it is to attach something to the tarantula? Obviously it's able to be done with the rattlesnake and the kangaroo rat, but a unique, I don't know if morphology is the right word, but its body has unique characteristics, and you got to consider if your sensor is going to change its behavior or not.
Absolutely. I think I would definitely start with this with lab studies first to make sure that the tarantulas could molt. So they need to shed their skin. So if you're gluing things to them, you need to make sure that they're going to be able to shed just fine. How heavy the sensors are. I don't remember the numbers, but there's often little rules that biologists will follow on how heavy certain things can be. Watching them over time in the lab, we can tell, is this tarantula behaving like it was before I put the sensor on it, or is it behaving like its neighbor that doesn't have a sensor? So I think there's a lot of lab work just to get this off the ground, but it would be so cool.
My name is Ryan Hanscom. I am currently a Ph.D. candidate in the joint doctoral program of evolution with San Diego State University and the University of California, Riverside.
And will that program result in like a project or a thesis, or what's the kind of milestone at the end?
Yeah, so I have a dissertation and several side projects. So I've already published multiple papers in peer-reviewed journals relating to my work, and that will, in the end, go to a dissertation. But the kind of end goal is to get more grants, more publications based on the work that I do in the realm of ecology and evolution.
And what sort of specific things do you research?
Sure. So I really consider myself a terrestrial ecologist, and I really specialize in herpetology and mammalogy. So I study reptiles and amphibians and mammals. And I'm interested in how anthropogenic disturbances can really affect their behavior and their ecology. So I do my work in the lens of climate change, land use change, and all this other stuff, and look at animal behavior, predator-prey interactions, population ecology.
When you say climate change, what are some of the variables of interest?
For us, it's very much actually temperature, because my main study species is a rattlesnake, and rattlesnakes are ectothermic or cold-blooded, right? And they eat mostly small mammals. So they're eating an endothermic prey item. And it's thought, from theory at least, that with increasing temperatures, this predator prey interaction between an ectotherm and an endotherm will be one of the first ones to be disrupted within the ecosystem. So there's all this other stuff to climate change, rising sea levels, all this other stuff, but temperature is at the focus of my work.
And you say it's a hypothesis, I guess, that it's one of the first areas to be disruptive. What kind of data you're gathering towards that end?
Yeah, so there's been a few papers really that have looked at it from more of a theoretical way and not looked at animals like on the landscape. They've just done it in the lab a few times and just using, physics and physiology. So right now we're not in the place that we're done with the work yet. So we've set up a system where we can test this in the natural world. And what I mean by that is there's two specific species. One rattlesnake, it's called the prairie rattlesnake, and it exists all the way from southern Canada to northern Mexico. Luckily for us, the prey item that it eats is a Oryx kangaroo rat, which is a certain type of kangaroo rat. We can talk more about these specific animals later if you'd like to. And that species geographic distribution also spans that range. So we're set up with a natural laboratory, really, a natural laboratory with an awesome thermal gradient all the way from Texas to Canada, where we can study the predator-prey interactions between two specific species. As temperature rises, performance changes, whatever that trait performance is. If it's a snake striking, that is immediately affected by temperature. Wherein as an endotherm, that kind of trait performance stays kind of leveled off. We're doing that via multiple things, and we haven't got to the end result yet where we can say, hey, as temperatures increase, it's more likely that the kangaroo rat populations will decrease, stuff like that. So that's what we're working towards.
So then in the agent-based approach, there must be some sort of model of the predator and prey. What does that come down to? What is in bits, what is a rattlesnake?
So in bits, what is a rattlesnake? This isn't the focus of my work. I kind of gather what makes the bits. So what is making the bits is stuff like the interaction rates between kangaroo rats and rattlesnakes, the successful feeding events, are they successful or not? The spatial behavior, spatial ecology of both of them. How much do they move? What are their movement rates? The population ecology of both species. How many individuals are there? So all of that is building, are the kind of the bits to the model, and is parameterizing the model. And then we add in all the temperature stuff and even more. I could go on for a while. So that's kind of the crux of it.
So it's all coming from the field, the natural environment. How do you collect your data?
We collect a bunch of different data, and I think it might be good for the listeners to kind of talk about what a rattlesnake is and what a kangaroo rat is. So, right, a rattlesnake is actually a pretty high density predator. You just don't see them a lot. It's a mesopredator. So what they do is they go out and they search for a spot in the landscape where they think they might come across a prey item. They'll set up an ambush. So they'll coil up and wait. They'll find they'll smell, use all their chemosensory ability to find a spot in the environment where they think it's a good hunting opportunity. And then they'll sit there and they'll wait for a long time, up to 24 hours to a week for something like a kangaroo rat to walk by. And after that, they strike at the kangaroo rat, and they're venomous. So they inject venom into the kangaroo rat, which is a small bipedal animal, and they're nocturnal. And then after that, the kangaroo rat, if the snake was successful with the strike, it runs away, it dies somewhere. The snake actually follows the scent of its own venom and goes and finds the kangaroo rat and swallows it. And kangaroo rats, they are considered a keystone species in our ecosystem engineer because they're building burrows throughout Western North America everywhere, providing habitat for other animals, and they are granivorous, so they only eat seeds. And what they do is they can change the whole vegetation landscape. It's been shown in studies before, if you exclude these animals, the vegetation community shifts drastically and quickly. That's kind of why we're interested in this specific interaction. And the way we collect our data is we use traditional methods such as mark recapture, where we mark an animal and we go out and try to find it again, and we get population estimates. Spatial ecology using radio telemetry, where we can track an animal, find where they are, see how they're moving across the landscape. But the really awesome one, which I'm very excited about, is we're using accelerometry now. So it's kind of a new tool in the animal behavior biology where we can attach a unit, an accelerometer unit, to an animal, validate behaviors, use a machine learning AI approach to create a model, and then I can let that kangaroo rat go. I can let the snake go. We don't have to watch it. It just goes out there and goes about its business. I get the data back, and then I can see what it was doing, and we can observe the unwatchable, which is really awesome. And it's a huge step in animal behavior because of how machine learning and AI is just getting better and better and better.
Very neat. I'd love to dig into all aspects of that. Are you ready to unlock new levels of success in your career, creative pursuits, and personal growth? The Hacker Mindset is a productivity cheat code to unlock new levels of success in career, creative pursuits, and personal growth. It's about leveraging the principles of hacking and applying those skills to the broader world. Available for pre-order now, The Hacker Mindset is not your typical productivity guide. Written by Garrett Gee, a seasoned white hat hacker with over 20 years of experience, this book reveals the secrets of hacking and how you can apply them to overcome obstacles and achieve your goals. The Hacker Mindset is a unique take on productivity, as Garrett took 20 years of white hacker hacking experience and identified the general principles, techniques, and lessons that anyone could use to overcome barriers and work towards their goals. What sets the Hacker Mindset apart is its unique perspective. It sheds light on the positive aspects of hacking, showcasing the good that can come from leveraging these skills. In a world where hacking often gets a bad rap, this book shows you the white hat side, the side focused on innovation, problem solving, and ethical principles. Pre-order your copy today at hackermindset.com/dataskeptic. Join the movement and embrace a new way of thinking. That's hackermindset.com/dataskeptic. Very neat. I'd love to dig into all aspects of that. I guess to start with, could we talk about the hardware? Is it something you can deploy broadly? Are they expensive units? And what exactly is going on the back of the animal?
Yeah, so these are piezoelectric accelerometers, if you know what those are. So basically, they are gathering acceleration data because there's a spring inside the hardware, and whatever way the movement of the unit goes, it produces a voltage, and you can kind of convert that into acceleration. You have one in your phone. Accelerometers have been around for a long time. But now they're becoming so miniaturized, and the battery life is becoming so much better, where I'm taking 25 hertz data on triaxial accelerometers on animals as small as 50 grams for 8 days. What's really awesome about this is that accelerometers and all technology is being kind of produced for other things and not to put them on kangaroo rats or snakes. The units are becoming smaller and cheaper. Faster. So we pay about $400 to $500 per unit we put on the animal, and we customize them. I don't do it myself. I work with a company in Europe, TechnoSmart, and they do all the manufacturing of these, and we can kind of make them the right size, for the perfect fit for our animal of interest.
And then how do you know that the presence of the sensor doesn't so upset the animal that their behavior is 100% different?
Ecologists in general have been putting things on animals for a long time. So we have kind of a threshold that we've developed throughout mammals and reptiles, where about 5% body weight is kind of what you want to keep the unit at, or less, for them to behave normally. But you always want to continue to watch and improve those animal ethics portion and make sure they're behaving normally. The best evidence for that is we've seen 2 kangaroo rats with backpacks, reproducing in the wild, right? We've seen snakes eating. They're doing things that they're normally doing. So that's really, you want to always continue to refine those approaches as much as you can, and we're on top of that.
And then, I mean, you've got to have not a perfect success rate of retrieval. How many do you get back?
Yeah, so I consider over the last 3 years, I've only lost three snake ones and I think two kangaroo rat ones, and we've collected data on over 50 individuals of each species. This is atypical. We
Wow.
usually lose a lot more of tags and loggers, but I think we figured it out kind of like because we attach a mini radio transmitter to the top of this unit so I can pinpoint exactly where it is if it falls off, if it's underground. So we're able to collect a lot of them. One poor kangaroo rat I think got eaten by a coyote, and because I saw tracks at the top of a hill and I never heard the signal again. So, but so we've lost about 5%, a little less than 5%.
Well, that's a pretty good rate, I would say.
Yeah, thanks.
And then I know accelerometer data is pretty raw data. Where do you get
Mhm.
from that towards something you're going to do analysis on?
These are huge data sets. It reminds me of working with genetic data sets when I was doing that during my master's, because I mean these CSV data sheets that we get back, like, right, you can't even open them in Excel if you wanted to. We're getting millions and millions and millions and millions and millions of rows of acceleration data. And what we do is we first have to manually annotate behaviors. So we'll set up in the field or in the lab, we'll watch these animals doing what they're doing. So we'll watch a snake eat a bunch, right? We'll watch kangaroo rats forage. And once we have that information, we can match up the acceleration signatures with the behaviors the animals are doing, and we can build a machine learning model. Once we have that machine learning model, which I've been using a software for, and I'm not building them myself even with code yet. I'm, like, right, like I'm prepping all of the stuff in Python, and I know you guys will hate to hear this, R, the program R. so,
I love R.
so Python. Do you like R? Okay, good, because a lot of computer scientists are like, "Oh my God, you guys use R?" I'm like, yeah, I just do. I'm an ecologist. There's a software online that actually like builds the model for you. And you can do random forest, different types of supervised machine learning models. So that's where the only from our lab group, that's the only things we've kind of explored yet. But people are doing unsupervised machine learning models, AI models with these type of data, because once I build that validation data set, then I can apply those. I can predict using that model the behaviors of the animal when I wasn't watching it, and say like, oh, it was eating all night. It ate on this day and stuff like that.
So in supervised learning, you need one column to be your objective, what you're trying to predict. What does that column have in your data set?
I think that would be behavior, right?
But how do you get behavior? It's not part of the accelerometer data.
Yeah, that's because we're watching. We manually pair that together. So in the lab or in the field, we take videos of animals, and we're like, oh, it's moving right now, and I know exactly what time. So we're making that fourth column manually by videotaping animals, and the behavior would be that column, and then you have the three acceleration columns, X, Y, and Z.
And what sort of accuracy do you end up with in the model you finally develop?
And it could be anything really. So sometimes it works really well. Sometimes it doesn't work really well. I can give you two examples. Let's talk about kangaroo rats first, where we built one model where we wanted to be able to distinguish between foraging, grooming, traveling, and motionless. Like those were our four behaviors of interest that we thought were very much ecologically relevant. The model was really good at determining what the difference between when the animal was still and when it was traveling. And it was really good at determining, we're talking like, above 90, like for F1 score, accuracy, precision, all that stuff. It was really good at doing traveling versus foraging and grooming. But for foraging and grooming, it kind of got confused. And that's because those are kind of similar motions. It's when the animal is kind of still but moving. So, like, they're putting seeds in their mouth or they're like scratching their head, but travel. So it kind of got confused between those two. But I mean, we're just still scratching the surface of this. Like, we're going to be able to build a lot better models too. So we're just, for some behaviors, we're well above 90%, and then other ones sometimes it's going to be around 50%. And we are always continuing to try to refine those models by adding more data. We're collaborating with other people to build specific models now. So yeah.
And then my understanding is that having validated against lab tests where you have a ground truth, you observe the animal, you know the model does a good job at predicting, so you can now predict on the raw accelerometer data from the field and get sort of a time series of what the animal did that day. Is that the ultimate outcome?
That's the ultimate outcome, and it's amazing because within the realm of behavioral biology, like that's something that's we've never been able to do in the past without this technology, right? Like from wanting to know what free ranging organisms are doing, the only way we used to be able to do that was first by just observing and writing it down using binoculars, to video cameras, to now to accelerometry. And it gets rid of a lot of those biases of like human presence, right? Like animals are scared of you. If it's a rattlesnake, doesn't want to see you, like, right? The only time you'll ever actually see a rattlesnake up and rattling at you is because there's a human there. They don't actually normally do that. So, like, this technology is like just providing us with a huge new kind of way of being able to quantify animal behavior of free ranging organisms. It's really awesome.
It seemed like almost a new door is open then. You can measure the actual day to day in the life of. Have there been any insights that have come out of that time series?
The snakes we're still working on, but I can really get into what we've figured out with the kangaroo rats. So there's, if you're not familiar, kangaroo rats are small mammals, right? And they're nocturnal, so they're only out at night, and we quantified that using accelerometry, so which was cool. But everyone's like, "Oh, they're nocturnal." Well, now I can like tell you they're nocturnal because we have data, not just anecdotal observations. But anyways, it's also thought that the moonlight will drive small mammals to not do as much. They'll kind of hide. They're afraid of the moon. If you've ever been outside at night and paid attention to the moon, you would know that you can actually see a little bit more when it's when it's a full moon. There's an idea there that these kangaroo rats or any small mammal, when it's a full moon night, they're in their burrow. They're doing the least amount of stuff as possible to come outside and be exposed to predators. Accelerometry gave us a perfect opportunity to ask that question. Be like, hey, are they really scared of the moon? And we found that no, they weren't. They weren't really scared from the moon. This is the best, most unbiased data we have. And sure, it's one population at one site. And this is the first time we've done it, but we're going to keep doing it. If anything, like, they were maybe when it was they were maybe more out when it was actually a half moon and when the moon was risen, because you have to take into account when the moon sets, when it rises, and all this other stuff. But we found that that didn't hold true, which was really, really awesome insight. And we also looked at different environmental variables and just to see like, hey, did temperature, humidity, other things, precipitation, stuff we can just measure pretty easily that we did, and something came out which was really interesting, and that we found that kangaroo rats would forage more when it was humid out. And we weren't even looking for this. And this just came out of using this technology that, oh, these are arid animals, right? They live in the desert, they live in an arid environment. We know they're worried about water loss. Are they actually behaviorally selecting to come out to eat when it's the most humid out so they don't lose as much water? It's amazing. So, like, stuff we've never even thought of before. And yeah, that's kind of some of the results we have for now.
On the rattlesnake side, I was surprised actually to learn from the paper something about their eating habits. Could you discuss that and what makes that challenging to study them?
Two of their behaviors last very, very different times. So the snake strikes, that happens in less than half a second. When the snake swallows a prey item, that can take up to 6 minutes. So it put us in this, and we're building a new model now too. But it put us in this position where we wanted to, we just had to build a classifier that only classified strike. Then we had to build a classifier that only classified swallowing. Yeah, that poses a problem because of window size. So we're trying to build one with like a rolling window size now and all this other stuff. But it worked pretty well in the lab, but we weren't able to get it like across species. And that could be due to body size, could be due to prey size that we were feeding them. So we're actually trying to add all that in together because in the end, what we really want to do is build a rattlesnake specific model across all species, many species of rattlesnake. We did it with 3 in this paper that you were referring to, and we couldn't really get to the final point of building a cross species model where we can just apply it to any of the rattlesnakes. And that's what we're working towards. And if anyone's interested in that stuff and wants to reach out to me, we're currently working with a few collaborators trying to build more models to make it better to be able to use just acceleration data set from any rattlesnake species of any size and figure out when they're eating.
Do you have any insight as to maybe what the weak link is there? I could see maybe something about the accelerometer because it's such a fast event. Maybe that needs to improve, or maybe the model needs to improve, or maybe every strike is a little different. What makes it challenging?
All of that. Like I said, I think body size also and prey size could really affect that, because you're going to have different accelerations for a small snake versus a large one, right? When the food item passes underneath the accelerometer, like it has like a bolus, if that's a really big food item, you're going to have a way different acceleration signature. So I think it's a matter of continuing to add data, optimize the models, and eventually we'll be able to get there.
Well, I can picture some sort of sensor on the Ord's kangaroo rat because they're kind of small, could fit in my hand. Where do you put it on this long snake?
Ideally, we would put it on the head of the snake because that's where all the action happens on a snake. But we can't. So we put them 25% of their body length. We put that when we standardize that across snakes, and we attach it just straight to their back using this veterinary bandage called tegaderm. And we literally just put it on the back of the animal, put on this veterinary tape, and it's lasted in the field for us for like 3 years now. It falls off sometimes, but it's done a pretty good job with the rattlesnakes.
Well, it makes sense to me that with the accelerometer data and the model, you could probably measure out behaviors that tell you if there's a feeding event taking place, like the strike behavior. You'd also mentioned having an interest in interaction rates. It's not clear to me that you can figure that out from an accelerometer. How do you know?
In terms of feeding events in rattlesnakes, that would technically be a successful interaction rate. If we have it on snakes in the field, we know how many times they eat, and we know a general size of a kangaroo rat. We can get into energetics there, right? And know like how many kangaroo rats do they actually need every month or something like that. We also used fixed videography in the field. We'll track our snakes. They're all have radio transmitters implanted in them too, and the accelerometer goes on the back and is an external attachment. We set up video cameras on them, and they sit in their ambush position, and we watch them all night long. And that's also how we gather some of this validation data for accelerometry. It's kind of a two-pronged approach there, where we can also figure out kind of the rate of successful strikes versus unsuccessful strikes. I think eventually we'll be able to validate successful strikes that hit prey items and strikes that miss them, because you would imagine that if a snake strikes and it hits something, there's going to be a fast deceleration, right? It's just going to like stop. And if it misses something, it's going to go, whoa, and like slowly bring its head back. So I think eventually we might be able to validate that. And we are working on that. We're always, when we do this stuff in the lab, we're grouping behaviors as detailed as possible. So in the future, hopefully we can have a model that might be able to figure that out.
Well, you talked about a pretty wide range for these two species. How do you decide where to go and do the deployments?
It's hard to do, right? You wish you could have samples and from all across the entire gradient from Canada to Mexico, but we've decided to choose 3 different sites, one at the very north of their range, one at the south of their range, and one right in the middle. So we work in Alberta, Canada, in Nebraska, and in West Texas. This is primarily where we're gathering a lot of this detailed behavioral population demography and ecological parameters.
And do you notice pretty clear differences between those 3 points?
Oh, yeah. Just to give you an idea, for body size is different. Snakes in the north are much bigger than the ones in the south. Same with the kangaroo rats. The snakes in Canada only have about 4.5 months of time they can stay active because they have to overwinter, while snakes in Texas have 9 months before they overwinter. We're finding that the kangaroo rats are kind of, in the same kind of deal there, where we think that snakes might be having effect on the kangaroo rats at the northern end of their latitude. So yeah, we're seeing some stuff, but we still haven't fully analyzed all. I wish I could give you a clearer picture, but eventually that will come out in the next few years.
And I know you guys already have some machine learning in the mix that we've talked about. What do you see as the potential for AI to help out your work going forward?
Yeah, I feel like that would be a great question for you. And like that, like how would you see after I explained all of this, kind of how we're using supervised machine learning, where you have a much, I just think that building an AI model might even be better if we could do that somehow, right? So yeah, I don't know, and I hope that we'll have the chat GPT of acceleration for behavior soon enough.
Sure.
Like, so yeah.
Well, with stuff like that, one of your challenges really is just the pipeline, getting that all running. I mean, what you described, I'm sure, was a monumental effort from many people. You got to get the data pushed into some clean training set. You've got to do the modeling, get that to work. There's many steps in that process. So that'll continue to be true, but some of that can get easier. I could see there being like a more generic modeling way that does the windowing you described and like auto figures out where to do it. Those kinds of technologies could be coming around and custom tailored to your use cases. Or maybe some boring stuff like figure out what the least intellectually demanding grad student is doing. Like if they're just labeling video, probably AI could do that.
Yeah, exactly. Right. So that's, yeah, that's what we want to happen eventually. Right. So I'm hoping that will come eventually, and it can only happen with us really field biologists, ecologists collaborating with, computation scientists. And that's what we've been doing. Yeah, I actually have a team of like undergrads that labels all my videos right now. So it'd be great if AI could do that.
Sure, yeah, under their training data, essentially. As soon as the machine can do as good as they can do,
Yep,
you can move them onto a more important project.
exactly.
What are some of the things in the future you're excited about? Where does the research go next?
For me, it really is kind of continuing to use technology in a biologging form to not just talk about accelerometry, like how can we use water sensors, light sensors to understand day to day behavior of animals. For example, working with this lizard right now in the desert, and something that it does is it buries underneath the sand. Sure, we can probably validate that with accelerometry, but if we had a light sensor, we could maybe also validate, when is it burying under the sand, because the light changes. I'm really interested in turtles as well. So, and specifically freshwater turtles, how if we have water sensors paired with accelerometers, paired with GPSs on these animals, where are they going? What habitat are they in? What are they doing? We just started, I think. Like it's all becoming so smaller too. So this accelerometry on animals really started with like big mammals, really charismatic animals, mountain lions. And the loggers used to be huge. You could never put it on a kangaroo rat. Well, now we're here where we can do this stuff. And it's really, really awesome. And it's just the beginning.
Do you have any advice for, let's say, undergraduates or young graduate students looking to get into some field research like you do?
For undergraduates looking to get into field research, I would definitely contact your professors and graduate students, and try to get involved as soon as you can. And everyone wants to make sure people have accessibility, especially me, to the field and getting into the field biology and having that opportunity. And in order to start, you have to make that initiative to go out to professors and graduate students and be like, hey, like, can I come out in the field with you when you're going? And start there and test it out. And then from there, you can understand if you love it. And then I was lucky enough during my undergraduate that I actually did like a whole project that got published eventually. So, I mean, I just reach out. Don't be afraid to ask professors and graduate students if they have an opportunity for you to help them in the field. That's a good way to start.
I think that's great advice. Can maybe be intimidating for some people. If you put yourself in their shoes, what do they have to offer? They're not an expert in the area yet. Is there time enough?
Yeah, I think there's a lot more. They could also ask about like volunteering at a local conservation group or something like that to get involved. That's something they wouldn't want to talk to one of their advisors or professors about. So there's all sorts of ways to kind of get involved with field biology, and starting as soon as possible in whichever way is the most accessible and the most comfortable for the student, I would highly recommend.
And is there anywhere listeners can go to follow you and learn more about your work online?
Yeah. I mean, I'm not using social media as much as I get to the end of my PhD, but I am on Twitter and I have an Instagram, so you can see there. But also check out ResearchGate, Google Scholar if you're interested in the type of work we do. I do in my lab, Dr. Rulon Clark, who's me and him together have developed this kind of stuff, right? Definitely reach out, and I'd love to talk more about this stuff with whoever.
Very cool. We'll have links to all the above in the show notes for people to follow up. Well, Ryan, thank you so much for taking the time to come on and share your work.
Yeah, I appreciate it, Kyle. It was nice meeting you.